three topics, which are somewhat related to each other. The first and second problems motivated us to tackle the third problem, MSA for prediction of antibody-antigen interactions. This problem is the hardest and today's main topic. The first topic is about our support for sequence analysis of coronavirus, especially for GIS8 database. Actually, this is an easy problem, and the changes we made was not heavy. We have been developing the MAFFT program for multiple sequence alignment since 2002. This plot is taken from our 20-year-old paper showing CPU time as a function of sequence length. For highly similar sequences, slope of this line, these two lines, is around 1, which means CPU time is almost linear to sequence length. But for divergent sequences, the efficiency is not so high. So MAFFT is efficient for globally aligning closely related long sequences, such as virus genomes of closely related virus genomes. So this program MAFFT is routinely used in this area. However, at first, we did not expect this application, global alignments of virus genomes, until we saw many analysis of coronavirus using MAFFT. After seeing these applications and receiving some questions and requests, we made some adjustments specifically for this application. This plot shows the number of citations to MAFFT papers, rapidly increasing since around 2020 due to spread of COVID-19. And here is a list of Nature papers related to coronavirus analysis using MAFFT. It's nice for us to see the contribution of our tool for solving this real-world problem beyond our original purpose. In this context, we recently made some small modifications to MAFFT as a response to requests from Dr. Raphael D. in GIS8. Today, I'd like to explain three options for constructing an MSA consisting of millions of coronavirus genomes. The results are available if you register GIS8 database with an option to download MSAs. Here is a screenshot to download the MSAs. Before asking us the request, they were using only a simple option of MAFFT, add fragments and keep lengths. First, I'd like to explain the simple calculation for MSA they originally used. This option is a combination of three command line arguments. First, the FFT algorithm is used. This is enabled by default. Second, by the add fragments flag, new sequences are added to reference alignment. In the case of SARS-CoV-2, Wuhan strain is used as reference. Third, by the keep length flag, insertions at the new sequences are all deleted to keep the length of sequence the length of alignment. As illustrated in this figure, if a new sequence has a deletion, new sequence is shown red. This is deletion in new sequence. Such deletion is included in resulting alignment and represented by gap. Such gap is here. On the other hand, if new sequence has an insertion like this, then the insertion is just deleted. This strategy works well in most cases, but sometimes this is not convenient because the insertions at the new sequences are always ignored. To avoid this problem, Raphael Lee in GISAID uses a bit complicated procedure like this. I cannot explain the detail of this procedure today, but in short, new sequences with insertions are listed up in advance. Next, the sequences with insertions are aligned to the reference sequence without deleting insertions, unlike the previous slide. So the alignment length gets longer at this step because of insertions. Finally, the remaining sequences are aligned to the alignment created by the previous step. As a result, without ignoring the insertions, an MSA with millions of sequences can be calculated in a reasonable time. This procedure is also convenient because they need to calculate only the increment of sequences, unless novel insertions are newly sampled. The detailed procedure is described in the README file in their data. He also asked me for a new option for codon-based alignment, and I newly implemented this option. Next, I'd like to explain this option and a limitation of this option. I think the usefulness of codon-based alignment is clear or obvious in this example. These are two alignments between Wuhan strain and mu variant. 3 bases, TAC, was inserted in the mu variant. At the nucleotide level, there is no difference in alignment score between these two alignments. However, the second one can be translated to protein more simply. So the second alignment is more convenient. To return this type of nucleotide alignment, considering amino acid translations, I newly implemented an option to codon pos and codon score. However, codon-based alignment does not always return correct alignment in terms of evolution. These two alignments are between Wuhan strain and alpha variant. The first alignment is quite simple. 6 bases, T, A, C, A, T, Z, G, were just deleted. Protein translation is a bit complicated because the position of gap does not correspond to codon boundary. Please see the amino acid alignment in black background here. 3 amino acids are unclear under this alignment. So the alpha strain is, in this case, HA, uncertain, gap uncertain, and S. On the other hand, in the codon-based alignment, this gap does not break codon boundary. As a result, amino acid alignment is clearer. Alpha variant has just 2 amino acid deletion, HD. However, the evolutionary event behind this codon-based alignment is a bit complicated. 2 events are necessary. GAT-GTC was deleted and A was substituted with C if accepting this nucleotide alignment. Considering this change occurred in a very short period of time, the first scenario is more likely. This problem is clearer in another case, the comparison between Wuhan strain and Omicron strain. Usually, the Omicron variant is described to have G142D and a deletion at the next 3 sites, 143 to 5. This notation is a bit confusing. No, not completely incorrect, but a bit confusing. I'll explain the reason. At the nucleotide level, the change in this region can be very simple. Just 1 deletion, as in this alignment. The origin of amino acid D, aspartic acid, is easy to understand. This aspartic acid was generated by a concatenation of originally different codons. Here and here. Guanine plus adenine cytosine. These bases were originally used to code amino acid glycine and tyrosine in Wuhan strain. Glycine and tyrosine in Wuhan strain. So this change was not a point mutation, but a deletion and concatenation of codon. On the other hand, using codon-based alignment, 2 different protein alignments are possible, here and here. In this alignment, upper one, valine tyrosine were deleted, and glycine was substituted by D. This is the same as the standard notation. However, this codon-based alignment, corresponding codon-based alignment, requires a bit complicated change at the nucleotide level. The changes are 2 transversion from guanine to adenine and thymine to cytosine, and deletion of 12 bases. The same problem occurs in 3rd alignment too. Amino acid alignment is simple, but it requires complicated changes at the nucleotide level. Considering these points, the first alignment is simpler and more likely than the last 2 in this case. Under this scenario, the notation G102D are a bit inaccurate or confusing. It can be misunderstood that G was substituted with D. More precisely, the aspartic acid was generated from a concatenation of split codon, not a substitution. Based on this consideration, they decided to provide both options, codon-based alignment and simple nucleotide-based alignment. In total, 4 MSAs are available in their site. I hope this data is widely used and helps many studies on coronavirus. The second topic is evolutionary analysis of coronavirus. We published in the last year, 2021, possible adaptive evolution in relatives of SARS-CoV-2 infecting bat and pangolin. I will skip this part because time is not enough. Here's our paper. Our conclusion is that relatives of SARS-CoV-2 infecting non-human hosts would have useful information to clarify the mechanism of infection. And probably we concluded probably there was adaptive evolution in outgroups, bat and pangolin, relatives of SARS-CoV-2 infecting bat and pangolin. This is, and I will move to the last topic. MSA for predicting antigen-antibody interactions. We are happy to see that MAFFT was used for analysis of coronavirus, as in the first topic in this talk, but I'm looking for other important problems for which MAFFT is not used yet. One of such problems is structure prediction of proteins. Recently, AlphaFold program by DeepMind achieved a great improvement in protein structure prediction. This program can also be used to predict structure of protein complex. This year, a new option, especially for complex prediction, was released. This option is called AlphaFold-Multimer. According to their paper, AlphaFold-Multimer also achieved a surprising improvement over conventional method. Here is a conventional method, and performance of AlphaFold-Multimer is shown here. The vertical axis is DocQ score, and I will omit the explanation of DocQ score. According to their benchmark, the performance was improved from almost incorrect to around medium by average. However, there seems to be a consensus that antibody-antigen interaction is difficult to predict, even for AlphaFold-Multimer. The preprint of AlphaFold-Multimer noted that AlphaFold-Multimer is generally not able to predict binding of antibodies, and this is an area for future work. We confirmed this result using some examples. This calculation was done by a doctoral student, Zi-Chan Xu, in our laboratory. In this table, each line shows an antigen-antibody complex with known structure of receptor binding domain of spike protein from COVID-19. And about half of these cases was successfully predicted by AlphaFold-Multimer, but remaining was not successful. This problem was relatively easy because only the receptor binding domain was extracted when running AlphaFold-Multimer. This result is more realistic and difficult cases collected from a wider range of antigens other than coronavirus. Only 3 cases out of 25 had medium or acceptable accuracy, and the prediction was incorrect in many other cases. The difficulty is easy to understand. In many protein-protein interactions, the evolutionary relationship is expected to be like this, ideally. In both protein 1 and protein 2, the binding sites should be more conserved than other sites, as there is a functional constraint to keep the interaction. Even if a change occurs in this area, a compensatory change can occur around that change. Probably such information can be included in training AlphaFold or other prediction programs. In antigen-antibody interactions, the relationship between sequence homologs are considerably different from usual ones. The relationship is kind of aggressive or unfriendly. For example, antigens sometimes escape from antibody. Also, even for the same pair of antigen and antibody, different epitopes in an antigen can be recognized by different antibodies. Finally, antibody can bind entirely different antigens. These antibodies are all homologous, nevertheless. Antigens are also homologous, even though they have different or no epitopes. So MSAs of homologous are calculated and used for prediction. But such MSA are confusing in terms of information on interactions. The MSAs can be mixture of antibodies that bind different antigens and mixture of antigens with different or no epitopes. A typical case was spike protein in non-human host, infecting non-human hosts. Based on this consideration, it's easy to explain why the prediction of antigen-antibody interaction is difficult. First, homologous sequences are generally used for predicting interactions. This is reasonable for many cases where those homologs share the same interaction and there is functional constraint to keep the function of interaction. However, this strategy is not expected to work well for antigen-antibody interactions. So it's not easy to solve this problem, but I'd like to briefly discuss what we are trying and planning, but I cannot present good solution yet. A key is to use a set of antibodies that bind the same epitope instead of a large set of antibodies. For this purpose, we are planning to use a new database called InterClone being built in our laboratory. This database works simply. A number of antibody sequences are stored as shown in this diagram. When you give an antibody sequence as probe, then similar antibodies in terms of CDRs are returned. This program only checks CDR complementarity-determining region. As a result, a set of antibody sequences that are expected to bind similar antigen is collected. Unfortunately, it's not easy to clearly distinguish antibodies that bind an epitope from antibodies that do not bind the epitope. This data is taken from a preprint using InterClone. This preprint is by Hendra Amant and co-authors in Osaka University. They have experimental data that was sampled from healthy donors and patients of COVID-19. Also, it's known which antibody bind an epitope and which antibody do not bind that epitope. This is known in their data experimentally. Using this data, an experimental validation of InterClone is possible. In this plot, antibodies in pink bind same epitope as the probe, and green antibodies do not bind that epitope. The vertical axis is the sequence identity to the probe, calculated from only the complementarity-determining region. So it's naively expected that green points and pink points clearly separate from each other. Indeed, there is such a tendency. Pink points tend to have a higher sequence identity around here. than green points. However, this tendency is not strong. Green points and pink points do not separate clearly. So the sequence identity at CDRs should be useful to some extent, but more data, not only CDRs, is necessary to clearly differentiate binders from non-binders. Using InterClone and other tools, especially for antibodies, we are trying to collect a better set of sequences, expecting that MSA of such sequences should be useful for prediction of antibody-antigen interactions. The last part is collaboration with Daro Standre, Chi-Chan Shu, Son-Lin Lee, and Hendra Isamot in Osaka University. Thank you for your attention.
Any questions before we break for lunch? So I actually have a question now. Your alignments on the variants. These better alignments actually can you somehow correlate into how severe these variants are? You know, we have a lot of data on how many people actually get infected by each alignment. Is there any predicted value that you can directly see as to how bad the virus will affect the population? So you showed the alignments of these COVID variants, right? Now that you have better alignments than before, predicts how bad this variant is going to be.
This is another step after alignment. Accurate alignment should be useful, I think, I hope, but the prediction of effectivity is another problem and probably a difficult problem.
Has not been tried based on your alignment?
I have not tried.
Any other questions?
Hi, I have a question on the AlphaFold for antibody-antigen prediction. So the solution you proposed using InterClone was essentially trying to subset the homologs to just the antibodies that bind a specific epitope of a specific antigen?
Yes, I expect that should work better than the normal AlphaFold.
Right. But in the case of a new antibody and antigen, you would need to know for a given sequence what homologs bind to the same epitope and antigen.
If the antibodies binding same homologs exist in database but does not recognize, then in such case I expect our proposal can improve the prediction.
I see. So you would need to have homologs that do bind the same epitope.
Yes.
And finding it is the difficult part, it seems like. To differentiate it between two other antibodies that might bind the same antigen but different epitopes or different antigens.
But that step is also difficult. We have not succeeded in clearly differentiate the binder and non-binders. Yes, this seems to be very difficult problem, but we are trying to solve.
I see. Thank you.
Professor Katoh, thank you so much for your talk. I have a slightly off-topic question. So in the beginning of your talk, you said it was possible to do the multiple sequence alignment at the SARS-CoV sequences these are longer than typical gene sequences, but they're not necessarily as long as some genomes. So my question for you is there a possibility that we can use your MAFFT software to do multiple whole genome alignment as long as there's no rearrangements? Do you think that's feasible?
Genome rearrangement is excluded from MAFFT calculation. So unfortunately, this is not, it's not possible to directly apply genome alignment. So it's necessary to split rearranged parts. And if the part without rearrangement is passed to MAFFT, then it may be useful. But it cannot be directly applied to the genome alignment.
Okay, so let's thank Professor Katoh again.
Yes.
Do we have lunch outside here or?
No, it's on, everybody's on, yeah, there is no lunch provided today. But there are many places around here that are actually quite nice. Well, there's. No.
More challenging. To have like these
Things that are hard to read.
Yeah.